Have you or a loved one ever been disintegrated by a Yellowstone geothermal feature? If so, you probably encountered the archaea Sulfolobus acidocardarius, or its relatives Thermus aquaticus, whose polymerase we use in PCR. Senecococcus, which gives grand prismatic spring its colorful rings, or thermoplasma, which also oxidizes salt. Sulfolobus are pleomorphic, meaning they come in many shapes. They are aerobic, hyperthermophilic acidophiles. They grow optimally in conditions with a pH of 3 and a temperature near 80 degrees Celsius. Organisms from the genus Sulfolobus have a thin S layer instead of a peptidoglycan found surrounding bacteria. While Sulfolobus stain gram-negative, gram staining is not a reliable test for archaea. Talk to your doctor if you want to know more about how to identify Sulfolobus. The species S. acidocardarius was first isolated by Thomas D. Brooke in 1972 from hot springs in Yellowstone National Park, and other species of the genus can be found in hot springs around the world, including Japan and Iceland. We are still learning more about these microorganisms. They grow around hydrothermal vents that spew hydrogen sulfide, so next time you walk off trail and smell rotten eggs, alert a ranger and don't jump into the hot spring.
Sulfolobus have multiple origins of replication. After a relatively short G1 phase, DNA replication occurs during the S phase. G2 phase takes up more than half of the cell cycle. During this phase, the cell grows and synchronizes with surrounding cells. This is followed by chromosome separation and cell division. Both are relatively short and have checkpoint inhibition.
In an extreme environment, do you suffer from an increased rate of mutations? Do you want the mutation rate similar to those microbes in normal conditions? How are they genetically stable at high temperatures and low pH? Well, your answer is protein. Thermostable proteins. Call the number on your screen and I will tell you how these proteins will help you succeed. Restriction endonuclease Sol recognizes methylation GC regions, which prevents cleavage and mismatching in nucleotides, which when fixing mistakes in the DNA sequence due to demyelination caused by high temperatures. Reverse gyrase will supercoil your DNA, preventing denaturation. Reverse gyrase is unique in that it is only found within hyperthermophiles, including Sulfolobus, and while bringing it to you. Call now and we'll throw in a non-specific binding proteins which attach and help prevent denaturation of the DNA. SAC7 is a DNA binding protein from S. acetylurus, which is not only resistant to heat, but also acidity. But wait, call now and I'll even throw in this cell membrane. This cell membrane is a monolayer made of lipids with ether linkages which are more heat stable and resistant to acidity. And if you're a virus, might I suggest the genus Sulfolobus. This Sulfolobus. Genus is a host for viruses already, especially S. acetylurus. Viruses such as the Sulfolobus icahedral turgid virus, STIV, replicate within the host and are protected from the harsh environment.
Breaking news. People are dying all around the world. The cause? Hydrogen sulfide. Hydrogen sulfide is flammable, corrosive, and denser than air. Besides disgusting people with its terrible odor, it is also suffocating people like the better-known dense gas, carbon monoxide. Why is there so much hydrogen sulfide? Well, the truth lies in tiny microbes from the genus Sulfolobus. The genus of organisms can oxidize hydrogen sulfide from their environment into sulfuric acid. They can be heterotrophic or autotrophic. In aerobic environments, they obtain their carbon from fixing CO2. When simple carbons are available in the environment, they can metabolize them and grow much quicker, making them speculative autotrophs. They're on strike and their co-workers, thermoplasma, cannot keep up with demand. Sulfolobes oxidize hydrogen sulfide at a higher temperature than thermoplasma, So less sulfuric acid is being produced, and our beautiful hydrothermal features around the world are less beautiful. The sulfur cycle is suffering. Also, people are dying. And now, on to Brian for our tech news.
Hello, we've got some interesting stuff for you today. Mining industries are always looking for ways to decrease their environmental damage, and archaea may hold the enzymes to do that. Their proteins are thermostable and acid stable and could potentially be used to remove harmful metals and acid from the operations. If you're not into tech, then maybe you're a scientist looking for a model organism. Sulfolobus is a model organism to study DNA replication in archaea because it is evolutionarily conserved and could be used to study the more complex replication in eukaryotes. It's highly transformable and synchronous in its cell cycle.